It's exercise 45. This deals with principles of heredity. As some background information on this, dealing with genetics essentially. For humans, in most of the cells, all what we call the somatic cells, which are the non-sex cells, there are 46 chromosomes. From a genetic standpoint, we would typically refer to this using the term as diploid, or for diploid, we often use the abbreviation 2N. Basically, what it means is you have 23 pairs of chromosomes. The egg and the sperm each have 23 chromosomes, so they would be classified as haploid or 1N, meaning they only have one copy of it. When fertilization occurs, the egg and the sperm will fuse together, and so the chromosomes of each now combine in the new nucleus. You would then go 23 from egg, 23 from sperm, and you're back up to 46. So you have 23 pairs. When you only have one copy, it's referred to as haploid or 1N. When you have two copies, it's diploid or 2N. So all of the cells in your body are diploid except for the egg and the sperm. So with that diploid number, like I said, that means you have one chromosome set from mom, one chromosome set from dad. There is some terminology that's used in genetics. Alleles is referring to the genes. And so on each chromosome, an homologous pair of chromosomes would be the chromosomes not necessarily exactly identical, but they contain similar information. One is from mom and one is from dad. So let's say you have eye color on it. Then you would refer to the alleles. That's the gene coding for the eye color. It is homozygous when both of those alleles are the same. It's heterozygous when they're different. So if you're talking about eye color, if they're both for, say, blue eyes, then they're homozygous. But if, say, from mom the gene is for blue eyes and dad is brown eyes, then it's heterozygous. They're both coding for eye color, but if they are expressed, it's different. The color would be different. When you have an individual then who is heterozygous for a particular trait, such as eye color, we also use the term dominant and recessive. Now, when you're heterozygous, the two alleles are different. So say for eye color, one is brown and one is blue. The dominant gene is the one that you would see. In the case of eye color, brown eyes is dominant over blue eyes. So if you have a heterozygous individual who has one gene for blue eyes and one gene for brown eyes, yes, they have those two genes. Yes, they are different, which is why they are called heterozygous. But what do you actually physically see would be brown eyes. And why is that? It's because brown eyes is the dominant gene. Blue eyes is the recessive gene. So you're not going, it's like it's masked, it's covered over. You don't see the blue eyes. It's there. It can be passed on to their offspring, but it's not what you're going to see in this particular individual. Typically, the only time that you will see one of these recessive traits, such as blue eyes, is if both of the alleles that are present in that individual are for that recessive allele. In other words, the only way you're going to see blue eyes is if both of the chromosomes have the gene on it for blue eyes. Because as long as one gene is for brown, you're going to see the brown eyes. So that dominant, if both alleles are for the dominant trait, then that's what you're going to see. In other words, if an individual has both of the genes are for brown eyes, then they're going to have brown eyes. If they're heterozygous, like we just said, in most cases, as long as you have one gene of the dominant trait, that is what you are going to see. A carrier is a term that we use when referring to someone who is heterozygous. Typically, we use this when talking about an individual that has a gene for a disease or some disorder or abnormality. They are not showing that disease or that disorder because they have a dominant gene, and that's the one that's going to be expressed, but because they have this recessive allele or this recessive gene, they don't develop that abnormality because the normal one is dominant, and that's what you're going to see. However, we call them a carrier because they can pass that down to their offspring. If both parents are carriers, excuse me, then basically they have they can pass that trait to their offspring. If their offspring were to receive, and it's purely by chance, if it was to receive the recessive gene from each of the parents, then they would develop the disease. And there's actually a 25% chance of that happening. As we continue with the genetics, a couple terms to keep in mind are genotype and phenotype. Genotype is basically, what do you see on the gene? Are they homozygous, meaning both genes are the same, or are they heterozygous, meaning they have one dominant and one recessive? It's what is on that DNA. The phenotype, think physical, is what you physically see. What color eyes? Do they have straight hair? Can they roll their tongue? It's what is being expressed. So it's what's being seen. So think phenotype, physical, genotype, gene. There are different types of inheritance. We're going to go over these. Dominant recessive inheritance is probably the most common one and what people initially will think of when you talk about genetics, but we'll also talk about incomplete dominance, multiple allele, and also sex-linked. For the dominant recessive inheritance, to set the background for this, a lot of the information was discovered by Gregor Mendel, who was an Austrian monk. He's considered the father of genetics. He did an awful lot of experiments with pea plants, and he was the one that finally was able to determine the way basically information is passed from parents to their offspring. He did find evidence that each gamete, remember those are the sex cells, the sperm and the egg, will contain just one allele, and then if fertilization occurs, the resulting zygote, which develops for humans into the embryo and then to the fetus, will have a pair, one from each parent. Now, he worked with what's known as monohybrid crosses usually, and we're just going to look at those for ease with this class. To figure out what a monohybrid cross is, basically what you're going to do is you're going to look at one trait. You are given the genotype for both parents, so you're given the information as far as for each parent, are they homozygous, are they heterozygous, and you're going to set up a Punnett square. This is an easy way of trying to predict what the possible genotypes for the offspring. So Punnett squares are usually used for prediction of offspring. It's not looking at past generations. You're trying to predict for future generation what the offspring, what their genotype is going to look like. If you know what the genotype is, then you're going to be able to determine what the phenotype is. So as Gregor Mendel worked with pea plants. He looked at several different traits. He looked at tall versus dwarf, purple flowers versus white flowers, wrinkled seeds versus smooth seeds. He had a lot of time on his hands. So if we look at an example that he did with the pea plant and looked at the alleles, or once again the genes, for whether the plant is tall or whether it's dwarf. Tallness is the dominant one. Dwarfness is recessive. He, through all of his research, was able to determine that. So in this example, we're going to use a cross. We're going to set up a Punnett square. And one of the parents is tall, and it is dominant, what you would call homozygous dominant. We tend to use letters. Just by tradition, for the letter you pick, you usually use the letter describing the dominant trait. So in this case, we're looking at tallness as dominant, so we're going to use the letter T. For a dominant trait, you're going to use a capital letter. For recessive, you're going to use the lowercase version of the letter. So we're telling you that one of the parents, and we're just going to call it the male, is dominant. It's tall, and it's homozygous, so it's big T, big T. You're going to cross this with another plant that is recessive. And remember, in order to see the recessive trait, you have to have both genes be recessive, because if there was a dominant trait, if it was heterozygous, it would be a tall plant. In this case, we're saying it's not. So it's little t, little t. So you are crossing a tall plant with a dwarf plant. When you set up a Punnett square, the main part of the square is going to be the lower 4 squares on the right hand side. That's where you're going, where all the arrows are. That's where you're going to be predicting what the offspring are. So across the top, you're going to label in the middle and the far right column with what were the alleles, the genotype of one parent. So, well, one parent in the previous slide we said was big T, big T. So in one of those blocks you put a big T, in the other column block you put a big T. On the left-hand side, you put the other parent, which was the dwarf plant, or little t, little t. So in the middle block, you put the little t, and then in the bottom block, you put the second little t. The letters in the columns bring those letters down, as you can see by the arrows. The little t's that are on the far left, you bring those rows across. So bring the letters and the rows across the columns down to fill in the 4 blocks, main blocks of the Punnett square. That is where now you have 2 letters, one from mom, one from dad, and it's giving you the genotype of, or possible genotypes of the offspring. So this is why you're using the Punnett square to predict what the offspring from this cross would be if you mated these 2 together. As you notice, for the potential offspring, all 4 of those blocks are big T, little t. So you would say the genotype, remember genotype essentially are the letters. It's what's on the chromosome. So genotype would be 100%, 4 out of 4, 100% would be big T, little t. And that is heterozygous because one is a big T, one's a little t. They're not identical. The phenotype, what you physically see, because it has a big T and that's tallness, and that's the dominant trait, that means 100%, 4 out of 4 would be tall. You can use, like I say, the Punnett square to predict what the probability of offspring will be for any of the traits. This is just another way of showing a similar thing, is that Mendel worked, as I said, with the flowers. Purple is a dominant. He found that when he crossed a purple flowering plant, pea plant with a white flowering pea plant, and for those of you who like flowers, I'm just going to say this, every species is different as to which color is dominant. For pea plants, the purple is dominant. So if you cross a purple flower with a white flower in that first generation, all of them will be purple, similar to what we just saw with the tallness. Because all of those offspring have the white flower gene, it's not being expressed because it's recessive, but it's still there. So it can reappear in future generations, as you can see with what's known as the F2 generation, that white flower can reappear. And some of you may be familiar with certain traits personally, maybe in your own families, of where you say, oh, something skips a generation. Why is that? It's often what you're seeing as a recessive trait. Incomplete dominance is where both of the alleles will be expressed in the offspring. So one's not completely dominant, as the name implies. If you want to set up a Punnett square, you do it the exact same way. So if one of those alleles is not completely dominant and you see expression of both alleles, what do you end up seeing if an individual's heterozygous? It's kind of an intermediate. The classic example is with snapdragons. Snapdragons do exhibit incomplete dominance. So you can have red flowers and you can have white flowers. If you cross a red flower with a white flower, what are the offspring going to have? Well, in this case, for the genotype, red flower is big R, white flower is little r, but it's incomplete dominance. So if you cross these, the offspring are going to be big R, little r. What does that physically look like? Well, it's going to be a combination of the red and white. And think of it this way. You mix red and white, what's halfway in the middle? It's going to be pink. And that is what you classically will see. So here's the crossing once again. You put one parent across the top, another parent down the side. Bring the columns down, the rows across to determine the offspring. Once again, the genotype 100% big R, little r. Phenotype 100% pink flowers. And here's another chart that is showing this. Once again, the P generation, that's going to be the original parents. The F1 would be their offspring. So like we just said, you cross the red snapdragon with a white snapdragon, you end up with a pink. It's incomplete dominance. It's both genes are being expressed. Now, if you were to cross the pink flower with the pink flower, then you end up with the F2 generation as their offspring, and on the very bottom, you can see where you would end up with, in predicting the probability of their offspring, a 25% or 1 in 4 chance of a red flower, a 50% or 2 in 4 chance of pink flowers, and a 25 or 1 in 4 chance of white flowers. Multiple-allele inheritance, oftentimes called codominance, means that several alleles may be expressed equally. The classic example for this is with the blood type. There's actually 3 different possible alleles, but each individual will only have 2 of those. The 3 alleles are the A, the B, and the small I. You'll notice the A and B are also written with a capital I because they are both dominant over the lowercase I, but they're also what we call codominant. If an individual ends up with a big IA and a big IB, both of those will be expressed. As I said, this is what we see with blood typing, that an individual who has type A blood, once again, you can only have 2 alleles. So you're either big IA, or you could be heterozygous. You could have the big IA, little I. An individual who is type B is big IB, or big IB, little I. An individual who is type O, that is the recessive expression, little I. If you are type AB blood, you have big IA, big IB. Both of those are being expressed. To relate this to the antibodies that are present and what type of blood you can get, this is a larger chart which is just showing that. Individual who is type O blood has anti-A and anti-B antibodies. They can only receive type O. Versus an individual, if you look at the bottom, who's type AB blood, they do not have the antibodies present, so they can receive all 4 types of blood. A person who is type AB is known as a universal recipient because they can receive all blood types. A person who is type O is known as a universal donor because they can give their blood to everyone. They can only get type O, but they can give it to everyone. Now, when we look at the 23 pairs of chromosomes, like I said, there's 46 total, but you pair them up because you get one from mom, one from dad. So there's 23 pairs. 22 of those are known as autosomes. That means they're dealing with the somatic cells. But the 23rd pair, this is the sex chromosomes. If you have an XX, then you're female. If you are XY, then you are male. There are some genes that are present on that X chromosome that we call X-linked or sex-linked. It has absolutely nothing to do with sex. It's just there's a lot of extra space on that chromosome. Some classic examples are color blindness, hemophilia, there's a male pattern baldness. So there's a lot of space there, but just because of the location of where it is, that it's on that X chromosome, we refer to it as sex-linked. This is a picture showing the difference in size of the X chromosome versus the Y chromosome. So you can see there's a lot of extra space on that X chromosome. Most of the sex-linked human disorders are due to recessive alleles. It's going to be more common in males than in females. And the reason for that is because males only have one X chromosome. The other sex chromosome is a Y. But because they only have one X chromosome, if it has that recessive allele for that disorder, they're going to express that disorder. They will have it. Females, in order to show a disorder that's caused by a recessive allele, they would have to have two recessive alleles in order for them to express it. Now, if they only have one, they would not express the disorder or disease, but they would be a carrier and could pass it on to their offspring. So if we look at color blindness, this is often depicted by drawing it as an X and then putting a C. Because color blindness is recessive, we use a lowercase c on that X. So it's showing that it's associated with that X chromosome. So females, the genotype could be both with big X's with big C's. They would have normal vision. They could be, as shown here, an X big C, X little c. That female would have normal vision, but she is a carrier. She could pass to her offspring that color blindness gene. The only way a female can be colorblind is if both of the X's would have a lowercase c on it. Males, there's only two choices. They only have one X chromosome. So that X, if it has a big C on it, they have normal vision. If it has a lowercase c, they are colorblind. And I'm just going to say here, males, because they get the X obviously from their mom, because that's all that females have are X chromosomes. So the Y came from dad, the X from mom. If a male is colorblind, that means that his mother is either a carrier or she is colorblind. Females, you have two X chromosomes. You got one from mom and one from dad. The only way a female is colorblind is she has to have both of those recessive alleles. She has to have both X little c's. Where does she get those from? One from mom and one from dad. If a woman is colorblind, that tells you automatically that her father is colorblind, or at least her biological father is colorblind, because the only way she's colorblind is to have the X little c's on both. And where did she get them from? As I just said, one from mom, one from dad. Dad only has one. So that means that if a woman is colorblind, her biological father must be colorblind. This is showing how you set up a Punnett square to determine, in this case, a sex-linked trait, in this case, color blindness. So we have the woman on top who is heterozygous, she's a carrier, and then the male along the side who is colorblind. Once again, bring the columns down and the rows across. So what are we predicting here? Well, obviously there's 50% chance of having a female, 50% chance of having male. For the females, there is a 50% chance in this situation that the female will be colorblind. There is a 50% chance that she will have normal vision. She'll be a carrier, but she'll have normal vision. For males, the offspring, there's a 50% that the male will be normal vision, and there's a 50% chance that the male offspring would be colorblind. This is just showing a pedigree chart. I just thought I would include it in here. Hemophilia is also something that is an excellent trait, just to show you as an example of how you can use a pedigree chart to look at past generations and kind of trace certain, in this case, disorders. If you start by looking at the bottom, Alexis, this is going through the royal family of Europe. Alexis, well, first let me back up and say circles are females, squares are males. If it's colored in, that means they have whatever disorder you are talking about, in this case, hemophilia. So Alexis, who is the son of Alexandria and the Tsar Nicholas of Russia, their son did have hemophilia. As I said, it is an excellent trait, which means he had to get it from his mother, Alexandria. Did she have hemophilia? No, she did not. That's why the circles only have colored in. She was a carrier. Where did she get her gene from? From her mother, Alice. She did not get it from her father. How do you know that? Was because if he had had the hemophilia gene, he would have had hemophilia, and he did not. So you know it had to come from her mother, Alice. Alice was a carrier. Where did she get her gene from? It was not, once again, from her father, because if it had been from her father, he would have also had hemophilia. So you know it came from her mother, who was Queen Victoria. So this is just showing an example using some history as to how to trace some of these genetic traits with disorders. Probability, you're just looking at the probability of how the chromosomes during meiosis in the production of the gametes, which are the sperm and the egg, which chromosomes end up in which cell. So you look at the number of specific events versus the total number of cases. So if an event is certain to happen, its probability is 1. But this is just showing an example of it. If an event happens 1 out of every 2 times, then it's 1 out of 2. And you can go continue on 1 out of 4. So oftentimes we're looking at the probability of, with genetics, of what the offspring, what's the probability of an offspring having a particular trait. It may be a positive trait, it may be a disorder that you're wanting to look at. What's the chances of maybe a couple's looking at, if we have a child, what's the probability of our child having a particular disease? And so this is an example of how they look at, if they know what the genotypes are of both parents, you can use the Punnett square and then determine what the probability is of an offspring having whatever the situation is that they're interested in.